Good morning, ladies and gentlemen. I'm Tomas Ryan. I will be chairing the first session, which is on the topic of neuroscience. In What is Life, Schrödinger asked, why would our brain, with the sensorial system attached to it, of necessity consist of an enormous number of atoms in order that it is physically changing and its state should be in close and intimate correspondence with a highly developed thought? 75 years later, we are no closer to having a good answer to that question about how to satisfactorily go from brain to mind, from the organ in our skull to the subjective conscious experience that we all experience every day. Schrödinger's What is Life led to the revolution in molecular genetics. We now understand how DNA codes for information, genetic information. We understand the genetic code, and it is now trivial to sequence the entire genome of any species on the planet. We know enough about genetics and molecular biology that we can be assured that in the medium-term future, cancer and infectious diseases will be a thing of the past. We have not had this kind of success yet in neuroscience. And for this reason, half of the speakers at What is Life, the Future of Biology, are neuroscientists who are addressing this question. They represent diverse subfields of neuroscience and different generations. Each has their own particular way of studying the brain, and each brings with them their own unique insights. All of them are, without a doubt, world leaders in their specific field. Some of them have contributed to recent technological revolutions in neuroscience. We now have the experimental tools for the first time through which we can make significant progress in understanding the brain. But we also need ideas. And the point of this meeting is to see, do we really have a good idea of how to understand the brain and how it leads to consciousness, to thought, to perception, and to memory? I can think of no better person to kick off this meeting than Professor John O'Keefe. Originally from New York and born to Irish immigrants, O'Keefe first studied philosophy at the City University of New York before doing his PhD at McGill University in Montreal on the subject of physiological psychology. That's what they called neuroscience back then. He then moved to UCL in London in 1967, where he has been working ever since. In 1971, O'Keefe discovered place cells. Amid the billions of neurons in the brain, place cells are neurons that temporarily become active when an animal or human is in a particular place. This is a foundational discovery for neuroscience and still drives much of the research in how we understand how the brain encodes space, how we navigate the world, and how we store memories. 7 years after discovering place cells, O'Keefe published a seminal book called The Hippocampus as a Cognitive Map. It has so far accrued over 10,000 citations, which is extremely impressive for an academic book, and it is still read by any serious student of neuroscience. The field that began with O'Keefe's discoveries and has been shepherded by him since then have led to the discovery of many of O'Keefe's original predictions. We now know about grid cells, border cells, head direction cells, and a whole set of different kinds of brain cells that create the picture in our brains of how we seem to be encoding the world. O'Keefe has received numerous honors for his discoveries, most notably the Nobel Prize for Physiology and Medicine, which he received in 2014 for the field and the discoveries that he created. Besides that, he has also received the prestigious Kavli Prize and honorary doctorates from the City University of New York, McGill University in Canada, and University College Cork here in Ireland. Please join me in welcoming to Dublin Professor John O'Keefe.
Well, thank you, Tomas, for that very fulsome introduction. Let me say how pleased and proud and honored I am to be here to join with you in this celebration of Erwin Schrödinger, who is one of my great fans, and to do so in the land from which my parents immigrated many years ago. I'm also pleased and honored to have shared the same platform with Mary Robinson, who is also one of my great fans, of whom I am a great fan. So, as you've already heard, Schrödinger was a Viennese physicist who was most famous for the development of his wave equation, which described the behavior of particles in the quantum world. He also, as you've also heard, had a lot to say about the genetic material and speculated on how, within the constraints of physics, such material would be constructed. He also, as you heard from Tomasz, said something about the brain, and I'll show you some of the quotes from some of his work, which I think laid out some of the most important questions which can be asked about the brain, and which, as Tomasz has said, we really don't have answers to yet. So I will speculate as to how we might be making progress towards those answers and where we're going. But as a caveat, when you're asked to talk about the future of anything, one of the most important things that should come to your mind is this famous quote from Yogi Berra, which is, "It's tough to make predictions, especially about the future." Okay, so here are some of Schrödinger's thoughts about the brain and the mind and some of the other aspects of the mind which are going to be difficult to fit into our current models of quantum mechanics and physics in general. So he said, I find it utterly impossible to form an idea about how my own conscious mind, that I feel to be one, should have originated by the integration of the consciousnesses of the cells, or some of them, and that's of course the problem, is it all of the cells or just some of them, that form my body, or how it should be the resultant of those activities. When we think that such a commonwealth of cells, as each of us would be the occasion par excellence for mind to exhibit plurality, if it were all able to do so. Why don't we have a multiplicity of consciousnesses? And this is, of course, the hard problem. Why is it that this brain, which we see as a multiplicity of cells, gives rise to only one conscious experience? He was, as many physicists are, a Kantian, and Kant set up what we now see as the framework, which shows how the world that's told to us by the physicists can be in some way mediated by our brain and our mind and our conscious experience, and what the relationship is between the two of those. So Kant said, and this is again Schrödinger, to be spread out in space and to happen in the well-defined temporal order of before and after is not a quality of the world that we perceive, physicists perceive, but perhaps to the perceiving mind, which, in its present situation anyway, cannot help registering anything that is offered to it except in these two coordinates, space and time. As I'll show you in a moment, Kant said that the only way we can perceive, given our mental equipment, the world out there, the physical world, is to see it as a spatio-temporal framework. The great thing about Kant, and this is of course important for physicists, was to form the idea that this one thing, mind or world, may well be capable of other forms of appearance that we cannot grasp and that do not imply the notions of time and space. And modern physics has gone down that route, and most of what physics talks about, quantum mechanics talks about, are instances and events which are not set in a spatio-temporal framework. Somehow or other, from this empirical evidence and from this empirical stuff, the mind and the brain make up our spatio-temporal view of the world. There is a problem about that, and one of them, and again flagged up by Schrödinger, is that the several domains of private consciousnesses partly overlap. My view of the world overlaps with yours, And where they all overlap is the construct of the real world around us. We actually believe that there is a world out there which is set in space and time. But if it's a construct of the mind, how does that come about? Prompting such questions as, is my mind the same as yours? Is there one real world to be distinguished from its pictures, interjected by way of perception into every one of us? And finally, he noted that consciousness is always in the present. We always perceive that we're acting in the present, in the now. Even one is remembering things from the past or predicting events of the future, they are viewed from the perspective of the present. Why should this be so? And how is this notion of the present now constructed? And I see these as actually some of the most important questions that are still facing us in brain research. We still have this Cartesian schism between our mind and our brain, between the brain that we study in the laboratory and the actual mind that we use to study it as we go about our daily business. So let me just say a little bit more about how Kant set up the distinction between these two domains. On the left-hand side of this slide is how Kant talked about the physical world, which he talked about as the Ding an sich, or the thing in itself. And whatever was out there, and Kant thought that we would never be able to understand or know about the Ding an sich, certainly was probably something which did not have a spatial and a temporal framework. And physicists now, in the latest, and I apologize for the physicists in the audience, but the layman's view of what they're doing, suggests that the way they see the world, either in string theory or loop quantum gravity, is as some set of some sort of a stuttering, frothing foam composed of tiny particles or tiny strings or tiny loops, which are continuously containing particles which come into existence as they interact with each other, then go out of existence to reappear at other locations in probabilistic locations. A very, very strange world, and one which I think, if physicists were actually honest, they would say we just can't actually picture this. So they have mathematical rules which tell them how it works, but they can't actually picture it. On the other side of this divide is our consciousness of objects set within a spatio-temporal framework, our ordinary consciousness of the world, where objects can only be perceived through the use of this framework. And so you can think of the mind and the brain as some sort of a filter, which takes this information from this world, inputs from this world, puts it to the filter, and creates the concepts of space and objects set within that space, and the relationships between objects. So space is created, as you can see here in this map of Dublin, and then within that are set objects, which might be inanimate landmarks or other objects, or animate objects, such as people, like Schrödinger. And then when we try to understand how objects interact with each other, then we create the notion of causality, something which had been actually spoken about by Hume, in which we say that if there are events such as pictured here, where an object moves in a certain direction, contacts another object, and then that second object moves, we see that as a causal relationship. But again, the suggestion is that there really is no causal structure to the world itself. So what I hope to do then is to sort of use this framework and talk about some work that's gone on in our laboratory and other people's laboratory to see whether we are making any progress towards seeing how the brain constructs this notion of a spatio-temporal framework and how it creates the notion of objects within that framework, and where we get the sense of the present now. How do we construct the present now, which we all experience as our moment-to-moment experience? Well, what we do in the laboratory is something very, very different from asking these questions. What we normally do is we look at the brain at a series of different levels. And they span from the angstrom level all the way up to the level of behavior at the meter scale. So at the lowest level, we talk about the brain as made up of individual isolated neurons. The Cajal doctrine of the neuron doctrine, which says that the brain is made up of individual elements which then communicate with each other, which poses, of course, the problem of where does this unified consciousness come from. We study the channels which cause them, the cells to respond to inputs and other events, and we look inside the cells and see what happens there. We then move up a level where we talk about the different individual neurons and the different cell types we find in different parts of the brain. Here's a drawing from Cajal, and you can see he's already started to picture lots of different cell types, and we can talk about the interactions between these cell types. So, for example, in this cross-section of the area that I'm interested in, the hippocampus, you can see that there are fibers coming from one set of cells which stream along into this area here and make contact with the cells there, which in turn project to the next area. So we think about the brain very much as an engineer would in terms of what kinds of transfer functions are created as you go from one stage to the next. And we have some good examples. For example, here in a cartoon from work by Jubel and Wiesel, where we can suppose that cells at one level, shown here, these 4 cells, which have very simple responses, for example, to spots of light, if they are then send that information to the next level, shown here, and the anatomy is such and the physiology is such that the cells that are sending the information line up in a particular pattern, they will create a cell which now is sensitive to lines. And you can think about going from one way station in the nervous system to the next as a way of transferring information from one representation to the next. We know from work, for example, fMRI and lesion work in neurological patients, roughly where things get done in the brain. We know that there's visual information at the back of the brain. We know that there are somatosensory and auditory information around here. And in the front of the brain, we think that that's where you do your thinking, your executive function. And we can look at behavior. We can train animals to make visual discriminations. We can put them into mazes and see if they can find their way around in the maze. And we can even make models of the human as they're walking around and study which parts of the brain, for example, the cerebellum, are responsible for that behavior. So to say a little thing about our progress in trying to understand this spatial framework that Kant talked about and that Schrödinger was so interested in, we know that a part of the brain called the hippocampus, shown here in the human and here in the rodent, is responsible for creating maps of the world. We call them cognitive maps. They're not restricted purely to physical space, but physical space and its representation of objects in the world and the relationships between them is, as shown in this map here, we think its primary function. And Adele and I, as you heard from Tomas, wrote about this in the late 1970s, and we still think that that's a very good way of thinking about this part of the brain. It does other things in addition, especially in humans, but this is a good basic thing. And we think, for example, that the simplest way of thinking about it is that there are places, and the idea of a place is actually an abstract concept which is created by the mind itself. There are no places in the physical world. But the places that the mind creates are then connected together with each other. You don't have a map if you just have a set of places. They're connected together by what we would call vectors, A, B, AC, CB, which tell each place representation where it is spatially relative to the others. To have a map, you need to have not only a notion about places, but you have to know about distances and directions. And over the years, following our original discovery of the place cells, we now have a fairly good idea of the whole panoply of spatial cells which represent different aspects of the outside. When I say represent, that's kind of a misnomer. They actually create the notions that I'm going to tell you about. So the place cells are interested in where the animal is, as shown by this heat map. So it only fires, the cell only fires when the animal goes to this location as it's roaming around this circular box. And it doesn't care, as shown in this polar plot, as to which direction the animal is facing in. By the way we do these experiments is we put animals into different shaped boxes, and we record from their brains, and we watch where they are with overhead cameras, and they have small lights on their head. But in addition to those place cells, we now know there are cells which are interested in the direction in which the animal is facing. Is it facing north? Is it facing south? And these cells are telling the animal about the direction between the different places. And finally, there are other cells, for example, the cells discovered by the Mosers, who got the Nobel Prize with me in 2014. Grid cells, which have this very beautiful pattern. They lay out a pattern across all the environments the animal is in, and they look like they're signaling, for all intents and purposes, distances in particular directions. So an animal would know how far it's gone in this direction by just counting the firing of these cells. Cells go brup, brup, brup. There are other cells which tell the animal how far away from the boundaries it is, so that it has some notion of landmarks. And you take all of these cells which are found in this area, the hippocampus, and surrounding areas, the entorhinal cortex, and that'll be important for a later point I'm going to make in my talk, and you put them all together. When I say you put them all together, it sounds like an easy thing to do. There are many, many computational neuroscientists who are spending considerable amounts of time trying to come up with mathematical models which say how these fit together, so that they, in the end, construct the idea of a place. Now, our understanding of these cells and what they do gives us a beginning of a hint of how this whole Kantian a priori spatial system might work. Because one of the things that my students, shown here, actually did was they tried to record from these cells in very, very young rodents, animals which were still in the nest or just leaving the nest. And what they found, to our surprise, although we shouldn't have been surprised as good Kantians, good Schrödingers, was that some of these cells, like the head direction cell, actually become active well before the animal's eyes are opened or well before it leaves the nest. So this is an animal which has had no spatial experience at all, and yet we can find in its brain fully fledged cells telling it which direction it's pointing. The other cells, the place cells and the grid cells, take a little bit longer to develop and probably are influenced in their development by the animal's experience. But here is, for the first time, good evidence that the spatial framework is there before the animal has any experience. So if you're going to have a spatial framework and you're going to create the notion of phrase, you want it to be there so that it informs your experience of the world. And this is probably partly or mostly laid down genetically or epigenetically. And we're still exploring this to find out exactly how this is done, but it is a very good system for studying the genetic basis of creating some of these fundamental ideas of which our brain is constructed. Now, one of the things that we've learned is that in many parts of the brain, particularly the cortical parts of the brain, the cells actually have to cooperate with each other, and they have to, the actual representation of an environment or a place in the environment is not held by one cell. It's not created by one cell. It's not represented by one cell. It's represented by a whole pattern across many, many cells. So we think of, for example, this group of cells in the hippocampus, which represent all these locations, as cooperating with each other and forming the representation of the space by cooperating with each other. You can't tell where you are by looking at just one cell. We used to joke in the 1970s about the grandmother cell. If you have a cell in your brain which tells you that's your grandmother as opposed to someone else's grandmother, it's almost definitely not the case. The cells represent bits of information, parts of information, then they cooperate with each other to form maps and such. That poses a problem. And it's posed a problem for us in neuroscience for many years now, because it means if we really are going to understand how the brain works, we have to record from lots of cells at the same time. And up until now, up until about 5-10 years ago, that has not been possible. But it's now becoming possible to do so because what we're getting in neuroscience is a flood of scientists from other disciplines, from nanotechnology, from physics, from chemistry. And they're bringing with them their ideas and their techniques, which are now enabling us for the first time to be able to record from large numbers of cells in behaving animals at the same time. So one of these techniques is one that our lab has been working on, and particularly Marius Bowser, and it really is based on computer technology. What it's based on is a probe which actually has many recording sites, 1,000 recording sites on a very, very thin shank, which is 70 microns by 20 microns, which we can insert into the brain, as shown here, and where we can record essentially from hundreds of cells at the same time. On that probe is essentially a computer. The probe analyzes the data, separates out the cells, multiplexes the information so that we can send it to a distant computer over very, very small lines. And we've used that to look at the grids, and we've been able to record from hundreds of cells at the same time, many of them grid cells, and see how they fit together and they talk to each other to actually create the notion of the animal's location in space. Now, I want to talk about this question of now. Where does this notion of now come from? And this has been actually flagged up by many people, and you might hear more of it in the conference from other speakers. How does the brain create this notion of now, which actually is synchronized with the events in this improbable physical world that I've described to you? Well, Sami Zeki at UCL has been one of the people who's thought about this the most. And he's realized that, for example, if you look at the signals coming into the visual system, the visual part of the brain, they come in at different times. So the signals that come in about the color of an object and about the motion of an object are actually arriving at very, very different times. And you can imagine that signals coming into the auditory system are coming at a very different time. So how does this fact that things come in at different times, how are they put together and projected backwards, because they're coming in late? If I'm sitting there looking at a screen and observing objects on the screen, I'm behind the real world. I have to be behind the real world. It takes time for all this information to come into my cortex. How does that work? And the answer is shown, I think we're beginning to get some idea of how it's shown, by looking at the cells in the hippocampal formation, in the whole system of cells, which create this notion of directions. So what you can then do is see where the cells get their information from, and we're talking about cells up here, which receive information from, among other places, the vestibular system, the balance system, so that when you turn your head, the system is saying, oh, you better update your representation of your heading. And this information, which comes originally from the vestibular system, moves up through a series of areas of the brain, going through the hypothalamus and the thalamus, and then to the cortex, where it begins to mesh with the information coming in from the outside world about how objects are moving as you change your direction. So you can tell your direction by either looking at the signals coming from your vestibular system, which tell you the movements that you're making, or information coming from the outside world. And what we find is that the information coming from your vestibular system, since it's actually part of essentially analyzing your motor behavior, leads the head direction, the real head direction, by almost 1/10 of a second. Then as it moves up to the thalamus, it only leads it by about 20 to 40 milliseconds. And then by the time it gets to the cortex, where it's now actually becoming integrated and synchronized with the visual information, it's at 0. So this creation of the idea of now is actually created by taking the signals from your motor system, signals that are already in your brain, your active brain, and putting them together with information coming in from your sensory system. So if it weren't for this fact that the brain is creative and active and always predicting what's going to happen, you would, in fact, lag behind the world that's out there. You would always be lagging a certain period of time behind the world that's out there. Let me tell you a little bit about how we look at this relationship between the information that's coming into the system, into the brain, from the sensory array, and how that's being put together with information which is actually coming from the animal's own movements. And one of the things we've been able to do recently is to create virtual reality environments for animals. For many years, we've been able to use virtual reality to study parts of the brain in the human, where we put their heads in a scanner and we look at which parts of the brain are active while they're watching virtual environments. But now we've been able to do this in animals as well. There's a video here which I won't show you. What we do essentially is we put the animal onto a moving ball, and we actually project images onto a screen in front of it. And as the animal runs on the ball, it perceives itself running down an alleyway. And on the basis of that, we can look at what cues are being used to inform the animal's location. We can look at how it's actually using its own movements to perceive its movement in space. And one of the things we can show is that these types of information actually interact with each other. So that if, for example, we have a cell which responds in the middle of the track here, if we take the cues away, then we see that the cell stops firing, and comes back again. And if we now do the following experiment, we can show that this cell is actually getting information from both the cues in the environment and the animal's own behavior. It's putting together these two streams of information. So, for example, we can put the animal down at the beginning of the track, and we can then allow it to see where the cues are, and then we can actually, as it starts to move down the track, we can turn off the cues so that it has no visual information about where it is. It only has its own motor movements to tell it where it is. And what we see there is that it actually, the cells can also use that integration of those two different types of movements to create this integrated notion of the world that we move in. How do we create this integrated perception based on all of these individual elements that we study in the laboratory? Well, we have been for many years taken with the neuron doctrine, the idea that cells actually are individual elements which operate independent of each other and talk to each other through chemical synapses. But there was another view, which was the view proposed by Golgi, who received the Nobel Prize early on, Wiskahal. And he thought, well, maybe this is not the correct view, that the nervous system actually formed what he called a syncytium or a reticular structure, where the cells actually were in contact with each other in some intimate way, either structurally, because they were part of the same big cell, or by the use of electrical synapses. And we're beginning to get information that in some way that there may be some aspects of that idea that are correct. It may be that the use of electrical synapses, which, for example, shown here, help to form oscillations in the brain. And one of the things that we're learning a lot more about is how the brain actually uses oscillations to synchronize information across various areas. As shown here, you can imagine that oscillations in the different parts of the brain here actually will cause the cells there to fire together and perhaps to communicate to each other through these electrical synapses. Okay, and the last thing I want to say is that we're now beginning to get the point where we think we know enough about the brain, and particularly structures in the temporal lobes that we've been talking about, to see if we can make some progress in understanding some of the neurological diseases which are the bane of many people's existence. And one of these diseases that I'm particularly interested in is Alzheimer's dementia, which is becoming, it now affects about 6% of the population of over 60s in the West, and will become a greater problem as we have an aging population. So how can we use our understanding of the areas in the temporal lobe and their role in spatial and temporal formation of information to help us begin to study Alzheimer's disease? Well, we can do it in two ways. We can look at animal models of the disease. We can create animals which have part of the disease. And we can also use it to create very simple tests which tax people's knowledge of the spatial relationships of objects in the world. And one of the important things about this is we want to have these tests so that we can see the earliest signs of the progression of the disease. Because if we're going to have a drug or some other way of intervening in the disease, we want to be able to do that as early as possible and certainly before there are the manifestations of the disease. So let me just show you one or two things. The disease, as we now know from work of Brock's, starts in this part of the brain, in the hippocampal formation here, and then spreads out along, we think, along nerve fibers to other parts of the brain. And the symptoms that people manifest are actually correlated with the spread of these toxic proteins, particularly the tau proteins, across the brain. So one of the things we've been doing, and our group in London has been doing, and my colleagues at Queen Square have been doing, is try to come up with simple spatial memory tests which will tax people who are at the earliest stages of the disease. And the test that they're using primarily now is called the 4 mountains test, where you show a picture of 4 mountains from a particular perspective, and you can see the mountains vary in height and shape. And then you ask the person, can you recognize these same 4 mountains amongst a set of foils from a different perspective? So you're looking at the mountains from this perspective, and now I'm going to show you the same mountains but seen from a different perspective. And in fact, these are the same 4 mountains here. It turns out that patients with damage in the hippocampus are very, very bad at this test. And when we look at patients who are at risk of Alzheimer's disease, who have what's called MCI, mild cognitive impairment, we find that they split up into 2 groups: those who are at risk and who are going to go on to have Alzheimer's disease, and who have already in their cerebral spinal fluid signs of damage, and they look very much like full-blown Alzheimer's patients, as opposed to patients who don't have those markers in their blood. So we're optimistic that using that we can begin to identify patients who are early at risk. And finally, I won't go into this, but we can use our knowledge of genetics and our understanding of this part of the brain to create mouse models of the disease. And when we do that and look at these place cells, what we find is that as these animals get older, some of them, not all of them, but some of them begin to show deterioration in the firing of these spatial place cells that represent the animal's location. So you can see here there's a cell that fires and tells the animal exactly where it is. But in some of these animals which we've created to mimic part of the disease, the cells are beginning to look very, very poor and not very good at spatial representation. So thank you very much for your attention. I think Schrödinger actually, in addition to giving us a blueprint for genetics, has also given us a blueprint for brain research. Because we've made tremendous progress in understanding the bits and bobs, the machinery of the brain, but we haven't made a lot of progress in seeing how that relates to our understanding of our conscious experience and our mind. We're getting new technologies, and I've told you about 2 of them. One which is the use of computer technology, and our friends from nanotechnology to tell us how to create probes which will enable us to record electrically from many, many cells in the brain at the same time. We can also use virtual realities, and there are other techniques which you'll hear about later on in the conference, where we can use optical techniques for looking at the cells in the brain and seeing when they become active, so we can correlate anatomy and physiology. And finally, I'm very optimistic. I didn't start in this field because I thought I would be able to contribute to mental and physical health, but it turns out that this part of the brain, which we've been studying for many of these years, actually is one of the first parts of the brain that gets involved and becomes disrupted in Alzheimer's disease. So we think we'll be able to use our knowledge to create mouse models and also to construct tests which are going to point to and identify people who are at the earliest stages of the disease and who would be suitable subjects for therapy. I've been very well funded for many years now by the Gatsby Foundation, the Wellcome Trust, who have combined to produce this institute in London now for studying neural circuits. Now that we're beginning to get the techniques for looking at neural circuits, we have built an institute, and I've been at University College all my life, as you heard, and thank you very much for your attention.